The term life cycle refers to all of the reproductive events that occur from one generation to the next. In animals, including humans, the individual is always diploid, and meiosis produces gametes, which is the only haploid phase of the human life cycle. While our bodies undergo mitosis regularly for growth and production of tissues, meiosis only occurs during gametogenesis. Gametogenesis is just the formation of gametes. For example, the production of sperm is known as spermatogenesis, and the production of, in females, gametogenesis is also known as oogenesis, or the formation of the ovum, which is also called eggs. We're going to go into each of these processes in a little bit more detail. Starting off with spermatogenesis, the testes contain sperm cells called spermatogonia. Now, these cells keep the testes supplied with the primary spermatocytes. The primary spermatocytes are diploid cells, which means they have how many chromosomes if they are human spermatocytes? So how many chromosomes would you expect to find in a human spermatocyte? These are then going to undergo meiosis to produce two secondary spermatocytes. These are now haploid cells, meaning that they have how many chromosomes? How many chromosomes would we find here in the secondary spermatocytes? The secondary spermatocytes are going to undergo meiosis too, and they're going to ultimately form 4 spermatotids. Now, these 4 spermatotids are genetically identical, or are they genetically unique at this point? These spermatotids must then differentiate, meaning that they're going to build up the necessary structures they need to perform their functions in fertilization. Once they undergo metamorphosis and maturation, they are called sperm. Oogenesis is going to be a little bit different. Oogenesis is different in the sense that it does not produce 4 haploid eggs at the end. Instead, only 1 egg is going to be produced, and at least 2 polar bodies are going to be formed as well. This is caused by an unequal sharing of cytoplasm during the cytokinesis. So we're going to start off with the oogonia, which is going to produce diploid primary oocytes. The primary oocytes are going to undergo meiosis 1 to produce 2 haploid daughter cells. Now, although those two daughter cells are both haploid, they do not have an equal distribution of cytoplasm. Basically, one daughter cell is going to get all of the cytoplasm and all of the organelles. So yes, the cell is going to grow in size just before division, but even though it grows in size, all of the bulk gets sent off to one of those daughter cells, and the other daughter cell is left with basically just a nucleus. So the large one will be called the secondary oocyte. The one that is much smaller and contains really only a nucleus is going to be a polar body. Then the secondary oocyte begins meiosis 2. Now, it doesn't finish meiosis 2, but it gets arrested at metaphase 2 to await fertilization. If it is fertilized, it will continue on through the rest of meiosis 2. If it's not fertilized, it'll just disintegrate and be sloughed off during the menstrual cycle. So here's a diagram that kind of shows what that looks like. The first thing that's going to happen, this is the whole structure that you see right here, is the primary oocyte. Like I said, it's going to grow very, very large. This all around it are regular human cells. So the primary oocyte is massive. And what's going to happen is that it's going to undergo meiosis 1. And when it does that, it's going to I'm trying to think of the word. I'm having a little brain fart. It's going to designate all of the cytoplasm and organelles to go to the secondary oocyte. The first polar body is essentially just a nucleus. There's very little remaining material left with it. This secondary oocyte is then going to undergo meiosis 2. During meiosis 2, it's going to be arrested at metaphase, and it's just going to kind of sit there and wait for fertilization before it continues on. Now, the reason for this, think about it logically. Why would we do this? Why would we want to have this process? Well, if you think about it, the sperm's primary contribution is DNA. The sperm's job is to be the first one there to deliver its DNA and carry on its genetic information. So there are lots and lots of sperm. Every turn of the cycle is going to produce 4 haploid sperm cells for every initial spermatocyte. And so the goal is to produce as many sperm as possible. We not only do we want a lot of them, but we want them really, really fast. We want them fast and efficient and streamlined so they can deliver their genetic payload as quickly as possible. So because of this, they are going to contribute very little to the zygote other than genetic material. Now, the eggs are only going to be released one at a time. They are precious cargo. There's only going to be 1 egg released per cycle and per menstrual cycle. And its primary job is going to just sit there and be ready to receive the sperm. As soon as it gets fertilized, it can start undergoing mitosis, can start forming that zygote immediately, and so it wants to have everything it possibly needs to be ready. So it's going to be very large in size. It's going to have lots and lots of cytoplasm. It's going to have everything it needs to begin very, very rapid mitosis as soon as it is fertilized. So the sperm and the egg that are produced by meiosis are produced through spermatogenesis and oogenesis. The sperm and the egg are going to fuse at fertilization, and this is ultimately going to form a single-celled zygote. So a zygote is simply the one-celled stage of an individual of the next generation. A zygote is just a single cell, but it has a complete and unique set of DNA, unlike any other creature on the rest of the planet. I mean, we've already talked about the odds of genetically identical individuals happening by sheer coincidence. It's not going to happen. So at the fertilization stage, at the zygotic stage, we have a single-celled, completely genetically unique individual. This zygote is going to undergo rapid mitosis. All of the growth from here on out is going to be mitotic cellular division to grow that cell as quickly and as fast as possible into an embryo and then into an infant. As a result of mitosis, each somatic cell in the body is going to have the exact same number of chromosomes as the zygote. It has the same genetic makeup, which was determined when that zygote was originally formed.